Okay, welcome guys. In this topic, we are going to discuss about the biosynthesis of urea, how the urea is synthesized. Now, the point is, when we see the word biosynthesis means we are going to synthesize urea in the body. The first question that should come in our mind is that why at all we require to synthesize urea in your body? What is the significance of that? What are the various clinical correlations that we have to find out based on these various pathways? So, first let's understand the need of the biosynthesis of urea, means why we need to synthesize urea in the body. So, when the need we can explain like this: when the proteins are catabolized, when we do the catabolism of the protein, means when we cut down the protein, the molecule that we are going to get is amino acid. And when this amino acid is further catabolized, ultimately we are going to make a molecule that is called as ammonia. This ammonia is a highly toxic molecule. Ammonia is a highly toxic molecule. So what we need to do is we need to convert this toxic substance into a non-toxic substance, that is urea. Urea is non-toxic. So we need to convert this toxic into non-toxic substance. Now this conversion takes place in the organ of liver. Liver has the enzyme to convert this toxic ammonia into non-toxic urea. Now once this non-toxic urea is formed, this urea is water soluble substance, so it can easily travel via blood. These urea molecule will easily travel via blood, will reach all the way to the kidney. This urea will reach all the way to the kidney, and in the urine this urea will be excreted out. In the urine via the kidneys, the urea will be excreted out. So this is the basic need of the biosynthesis of urea, that when you cut the amino acid, you will get the ammonia, which is a toxic molecule. We need to convert that into non-toxic form so that it can be excreted out of the body, and the conversion of ammonia to urea takes place in the organ of liver. This is the broad outline. This is the broad outline. Now we are going to see that how these various things or various steps are going to occur. We will see the micro details of that. So to understand the micro details, what I can say is, see, to understand the micro detail, I can put the things in this way. Say for example, there are various amino acids which are there in the tissue, and we want to catabolize them, right? So what we do is, first we convert all the amino acid into glutamate. Now, why we are doing that? See, the ultimate pathway that you have understood by this simple diagram, what I am saying is, what you need to do is you need to send the ammonia somehow to the liver. If your ammonia reaches to the liver, the ammonia is ability to convert into urea, and the liver is the only organ which has this capability of converting ammonia into urea. The liver is the only organ. So what we need to do is we need to send the ammonia in the liver. How we are going to achieve that? Whatever amino acid we have in the tissue, we want to catabolize them. First we convert them into a specific type of amino acid. And the name of that amino acid is glutamate. First we convert all the amino acid into glutamate. Because we are converting into one amino acid into another amino acid, because glutamate is also amino acid, so we have the 22 types of amino acid. So what we are going to do is, most of them we will convert them into glutamate, because we are converting one amino acid into another amino acid, and this process is referred as transamination. So the step 1 in the biosynthesis of urea is going to be, we will converting all the amino acid into glutamate. You will understand that why we are converting into glutamate when we will discuss the step 2. So now this glutamate is an amino acid. We know that it can easily travel via blood because it's a non-toxic substance. It is amino acid. It can easily travel via blood. So it will travel via blood and it will reach all the way to the target organ, and that is over liver. So say for example, here is the liver. So this glutamate molecule will reach here in the liver. Once the glutamate reaches in the liver, this glutamate has a very specific quality, we can say, that it very easily donates the ammonia. Means you can take out the ammonia very easily from the glutamate. So when you remove the carbon dioxide, I told you in the previous session that when you remove the carbon dioxide, that is called as decarboxylation. If you remove the ammonia, that will be called as deamination, right? And this deamination that is occurring from the glutamate, it occurs in the presence of oxygen. So this is referred as oxidative deamination. This is referred as oxidative deamination. This is the step number 2. The reason we have converted all the amino acids into glutamate is because glutamate easily donates the ammonia, and this is what we want. I told you that somehow you have to load your liver with the ammonia. If you are able to achieve that, the ammonia will be converted into urea. So how to load the liver with ammonia is because we know that glutamate has a quality to donate the ammonia very easily. Rest of the amino acid does not do that easily. So what we do is we convert first amino acid into glutamate. The glutamate will go to the liver and will donate their ammonia, right? So this is the process. This is the process by which we can load the liver with the ammonia. Now, there are some reactions which can generate trace amount of ammonia directly in the tissue. There are very few reactions, but the free ammonia can be generated in the tissue. Now, we need to transport this free ammonia all the way from the tissue, means from the extrahepatic tissue all the way to the liver. Now, how to do that? We cannot transport the ammonia directly via blood. The reason is it's a very toxic substance. If it comes in the systemic circulation, the clinical manifestation will appear of hyperammonia. So what we need to do is we have special transporters. We have certain special transporters. Now what these transporters will do? Different tissue will have different type of transporters. These ammonia molecules which are there, they will be packed via these special transporter in such a way that this ammonia will not remain toxic. Now via these various transporters, these ammonia molecule will reach all the way to the liver, right? So the step number 3 I can say is the various type of transporters that are there to help in the transport of ammonia. Now once you have loaded the liver with ammonia, what is the next and the fourth thing that we need to do is we will convert this ammonia all the way into the desired product, that is urea. Convert the ammonia into urea, and that is going to be the step number 4. And this step 4 that is going to occur, it is going to occur via the urea cycle. Once you have made the urea, we know that urea will go all the way to the kidneys, and will be excreted out in the form of urine. So I can say that these are the 4 steps. What I am saying is, first of all, you will convert all the amino acid into glutamate, right? That is called as transamination. The reason is because glutamate easily donates the ammonia. So this glutamate will go all the way to your liver and will donate the ammonia. That is called as oxidative deamination. So transamination, oxidative deamination, that is very straightforward. Then the third thing is there are some reactions in the extrahepatic tissue and they may generate a trace amount of ammonia, and that is supposed to be transported to the liver. And how to achieve that? We have special transporters. So that will be step 3, that is transporters. Now once all the ammonia is reached in the liver, we need to convert that into urea, and that is going to be the step number 4, right? The conversion of ammonia to urea, and the process that is called as urea cycle. So these are the 4 steps that are there by which you can convert the ammonia, that toxic substance, all the way into urea, right? There is some additional information that I want to add here itself in this flowchart, because in this entire session we are going to discuss the details of these 4 steps only. So here I want to add one more additional point. If I ask you that by looking at this flowchart, you have understood that why we have converted everything into glutamate, because all the amino acid into glutamate, because glutamate donates the ammonia easily. If I say that apart from glutamate, there are certain other amino acid also that can also donate the ammonia easily, then there is no need of doing the transamination for those amino acids. Those who can donate their ammonia directly, they can directly come to the liver and donate their ammonia, right? So there are certain amino acids, there are 3 amino acids which will not undergo transamination, which will not be converted into glutamate, because they also have the property to come to liver and to donate the ammonia, right? So I am writing that there are 3 amino acid, that is threonine, proline, lysine. These are the 3 amino acids. What they will do is they can directly come to the liver without any transformation. They can directly come to the liver and they can undergo oxidative deamination. They can directly come to the liver and they can directly undergo oxidative deamination and will donate their ammonia. So if they ask that which amino acid will not undergo transamination, then the answer will be threonine, proline, lysine, because they also have the capacity to donate their ammonia, right? So now we are going to discuss the details of these four steps. So starting with the step number 1. Step number 1. The step 1 that is there is the transamination. If you have understood the flow chart, then it is going to be very easy to understand these 4 steps. Whenever we discuss any reaction, the goal or the agenda of the reaction should be very clear. What is transamination is? Transamination means what we are going to do is we will have the different amino acids. We will have the different amino acids, and ultimately what we are trying to do is we are trying to convert these amino acid into glutamate. And the reason I have explained you that the step 2 is easy with the glutamate, right? So the idea is the substrate is going to be different amino acid, the product is going to be fixed, and that is glutamate. That is the agenda. When it comes to the location, the place, it occurs in all the tissue. It occurs in all the tissue. In this reaction, we do not generate any ammonia. Ammonia is generated in the step number 2. We do not generate ammonia in the step number 1. In the step number 1, what we do is we convert every amino acid into glutamate. So I can say no free ammonia is generated. No free ammonia is liberated. We do not generate any free ammonia. Then when it comes to the enzyme, the enzyme of transamination is transaminase. The enzyme of transamination is transaminase, sometimes also referred as aminotransferase. Transaminase or aminotransferase is same. The coenzyme is very important. Coenzyme is very important, and that is the active form of vitamin B6. In the decarboxylation also we have discussed the same. The decarboxylation of amino acid, we require vitamin B6 active form. Here also we require vitamin B6. The name of that is pyridoxal phosphate. Pyridoxal phosphate, which is written as PLP. PLP, pyridoxal phosphate, PLP. So in the transamination, we are going to convert the various different types of amino acid into glutamate. Let's see the examples. So, how we are going to do that? If we talk about the example, first example I am taking is aspartate amino acid. For example, if I have the aspartate amino acid, I want to do transamination, means I want to convert this molecule into what? The product is fixed, that is glutamate. Glutamate is going to come there and there only, is going to come, right? In order to do that, what we do is we bring a molecule that is with the name of alpha ketoglutarate. Alpha ketoglutarate. This alpha ketoglutarate is basically, it is a alpha keto acid. It is a alpha keto acid. So what we do is we transfer the amino group of aspartate on the alpha ketoglutarate. If we transfer the amino group on the alpha ketoglutarate, we will get this molecule that is called as glutamate. We will get the glutamate. Along with the glutamate, we will get again one more alpha keto acid, and the alpha keto acid that we are going to get is oxaloacetate. Oxaloacetate, and that is also alpha keto acid. So when you take the aspartate amino acid, you transfer the amino group on the alpha ketoglutarate, you are going to get the glutamate amino acid and the oxaloacetate, which is also alpha keto acid. So if they ask that what is the name of the enzyme for this reaction, as I told you, the name of the enzyme is going to be transaminase. The name of the enzyme is going to be transaminase because we are doing transamination reaction. The full name of the enzyme is, for this particular reaction, the full name of the enzyme is, now pay attention and follow the green color marker. The full name is serum glutamate, G stands for glutamate oxaloacetate transaminase. That is also referred as SGOT, serum glutamate oxaloacetate transaminase, right? The coenzyme, we know that it is going to be vitamin B6. It is going to be vitamin B6. Serum glutamate oxaloacetate transaminase, and the coenzyme is going to be vitamin B6. This is one example. This is the first example, how the aspartate converts into glutamate. Let's see the second example. That is how the alanine amino acid will convert into glutamate. This is the second example of transamination. How the alanine amino acid will convert into glutamate. Again, we are going to require alpha keto acid for the same. We are going to require the alpha ketoglutarate, that is alpha keto acid. And as a product, again, we are going to get the glutamate along with pyruvate. The pyruvate is going to be a byproduct, and that is also alpha keto acid. That also belongs to the family of alpha keto acid. So you are going to get pyruvate. When it comes to the name of enzyme, it is going to be, you can follow the green color marker, serum glutamate pyruvate transaminase, SGPT. Serum glutamate pyruvate transaminase, and the coenzyme is vitamin B6. The coenzyme is vitamin B6, right? So this is how we can convert one amino acid into another, and that is aspartate to glutamate, alanine to glutamate. So to summarize the transamination, this transamination is a very important reaction. If you ask me which is the most important reaction in amino acid, is the transamination reaction. We'll see there are so many clinical correlations are there. So what we have done, did in the transamination is, we had one particular type of amino acid. So I am writing, say for example, amino acid number 1. What we did is, we will use the alpha keto acid number 1. We'll take one alpha keto acid. I am writing number 1. We will use these two. They will react with one another. What we are going to get is glutamate, that is the amino acid number 2. We are going to get a different type of amino acid, so I am writing amino acid 2. And you are going to get a different keto acid also, so I am writing alpha keto acid 2. So this is what we are doing in transamination, that we are taking one amino acid, we are taking alpha keto acid and converting into a different type of amino acid and a keto acid. So this is what transamination is all about. This is how the transamination occurs. The point is how to remember the various examples of transamination reaction and what are the clinical significance of knowing all that. So first, how to remember this? We are going to use a mnemonic. We are going to use a mnemonic for that, and the mnemonic that we are going to use is SO LP. This is the mnemonic that we are going to use to remember the transamination. As you know that the glutamate is going to come in all the reaction. The product is fixed, glutamate. Even the bisubstrate is also fixed, that is alpha ketoglutarate. It is going to come in all the reaction of transamination. So there is no point in remembering that the glutamate is going to be product in every transamination. Even the alpha ketoglutarate is going to come in all the bisubstrate part. So how to remember the remaining things? So when it is aspartate, S, the byproduct is going to be O. So it is SO, aspartate oxaloacetate. When it is alanine, that is L, the byproduct is going to be P, that is pyruvate. So the mnemonic comes out to be SO LP. So S stands for aspartate, O stands for oxaloacetate, L stands for alanine, P stands for pyruvate. So when it's aspartate, oxaloacetate, alanine, pyruvate, right? This mnemonic is very important. Right now it's looking that why we are discussing so much about this mnemonic. You will understand when we will discuss the gluconeogenesis, various clinical correlation, when we are going to figure out here, then you will understand the importance of this, right? So LP, aspartate oxaloacetate, alanine, pyruvate, and this is how the transamination reaction occurs, right? Next, what is the clinical correlation of knowing this SGOT and SGPT? See, SGOT and SGPT are the component of liver function test. SGOT and SGPT are component of liver function test. You may have seen in the reports SGOT, SGPT, bilirubin, albumin, they all are component of LFT. So what I can say is, when it is a liver disease, whenever it is a liver disease can be divided into two categories. It can be alcoholic liver disease or it can be non-alcoholic liver disease. Broadly, we can classify like this: alcoholic liver disease, non-alcoholic liver disease. If it is alcoholic liver disease, what you are going to notice that the SGOT will be twice higher than the SGPT. SGOT is going to be twice higher than the SGPT. Whereas in the non-alcoholic liver disease, the SGPT is going to be twice higher than the SGOT. Twice higher than the SGOT. So let's say there is a patient comes to you with a report of LFT. The SGOT and SGPTs are on the higher side. Let's say one is 100, one is 150. They are on the higher side. So what you need to see is that which one is more higher. If it is SGOT which is more higher than the SGPT twice than normal, means the ratio is more than 2:1 or more, right? If it is like that, then you will say it is going towards alcoholic liver disease most commonly, most probably. And if the SGPT is higher, then it is going towards the non-alcoholic, say for example, the viral hepatitis or the autoimmune hepatitis. It can be like that. So this is a rough idea that you can get from the SGOT, SGPT. What are the other names of SGOT and SGPT? SGOT is also called as, see, SGOT, the SGOT we have discussed that it is for the transamination of aspartate. SGOT is for the transamination of aspartate. So it is also called as aspartate transaminase or aspartate transaminase. So it is also called as aspartate transaminase. In short written as AST, aspartate transaminase. Whereas SGPT, it is for the transamination of the alanine. So it is also referred as alanine transaminase. Alanine transaminase, so written as ALT. So they can use the word AST, ALT, that is just the other names of SGOT and SGPT, right? So this is how the step 1 is done. Step 1 is done. Here I can add the point that which amino acid will not undergo transamination, means which amino acid has no requirement to convert into glutamate. I have already explained in the flow chart there are 3 amino acids. It is not IPL, it is TPL, threonine, proline and lysine. Threonine, proline, lysine, there is no need of doing transamination. They will directly go to the liver, will donate their ammonia. So let's write down this point that threonine, proline and lysine does not undergo transamination. And the reason I have already explained to you. That does not undergo transamination. The reason I have already explained to you. The step number 2. Then is the step number 2. The step number 2 is to do the oxidative deamination, right? So step number 2 is oxidative deamination. Oxidative deamination. The reason why it is called as oxidative deamination, deamination means removal of ammonia, and oxidative means it occurs in the presence of oxygen. So oxidative deamination means removal of ammonia in the presence of oxygen. Right? We know that oxidative deamination is going to occur in hepatocyte, but if they ask in hepatocyte where, is it in the cytoplasm or the mitochondria? It is going to occur in the mitochondria. So we'll write down the place. Place is going to be liver mitochondria. Oxidative deamination is going to occur in liver mitochondria. Let's see the reaction part. We know that the substrate is going to be glutamate because glutamate was the product of the first reaction. So what we are going to do is we are trying to remove the ammonia part. We are trying to remove the ammonia from the glutamate. When you try to do that, what you are going to notice that in this reaction, NAD comes into action and converts into NADH. NAD converts into NADH. Now, this one point you have to keep in mind. This is one of the thumb rule for biochemistry, that Whenever there is NAD, NADH in any reaction, what we need to do is, the name of the enzyme is going to be dehydrogenase. Right? Whenever there is NAD, NADH reaction coming, the name of the enzyme is dehydrogenase. So the name of enzyme here is glutamate dehydrogenase. And the glutamate will take out the ammonia, right? So we will get the ammonia. Along with that ammonia, you are also going to get the alpha ketoglutarate. That is the byproduct. But what is more important for us right now? The more important molecule that we are primarily concerned is the ammonia. So this is how you have got the ammonia in your liver, right? This is the oxidative deamination, right? That is going to occur. What are the things to be remembered? Going to occur in the liver hepatocyte mitochondria. NAD, NAD is coming into reaction, so it is going to be dehydrogenase, and the ammonia is going to accumulate in the liver. Then comes the step number 3. In the step number 3, we are going to discuss the transport of the ammonia. The transport of the ammonia from the various tissue. Transport of ammonia. So from where to where we are going to discuss the transport? From the extrahepatic tissue to the hepatic tissue. The transport that we are going to discuss is from the extrahepatic tissue. We will start the journey from the extrahepatic tissue. It can be muscle, it can be brain, it can be any extrahepatic tissue, and will go all the way to the hepatic tissue. This is what we are trying to achieve. From the extrahepatic tissue, we want to go to the hepatic tissue. So to achieve that, let's say the first example is from, for example, transport of ammonia from skeletal muscle to liver. Transport of ammonia from skeletal muscle to liver. Let's see how we are going to do that. How we are going to transport the ammonia from skeletal muscle to liver. So to understand that, I am just giving you a diagrammatic representation. For example, this is the skeletal muscle, and here is the liver. And these are the free ammonia molecules which are there in the, I want to transport that all the way to liver. How to achieve that? We know that the skeletal muscle uses glucose for the energy. And how they conserve the energy, how they use the energy is by breaking the glucose. When they break the glucose, the molecule that they get is pyruvate. And this process is referred as glycolysis. So when we do the glycolysis, the glucose will convert into pyruvate. Then this pyruvate, sometime what it can do is, it may club with the pyruvate molecule. The ammonia may conjugate with the ammonia if it is available, and it will make a molecule, and the name of the molecule that is going to generate is with the name of alanine. Alanine is not a toxic molecule. It can easily travel via blood. This alanine can easily travel via blood and can reach in the liver. Once this alanine molecule will reach in the liver, it will again bifurcate into the two parent molecules. One is the ammonia and another is the pyruvate. So you can see that we have transported the ammonia molecules from the skeletal muscle to the liver via the alanine. The transporter name is going to be alanine. Then this pyruvate, what is going to happen with the pyruvate? It is may convert back into glucose, and if you synthesize glucose from pyruvate, this is referred as gluconeogenesis. And this glucose can be sent back all the way to the muscle for the energy purpose via the blood. Via blood, this glucose can be sent back. So this entire cycle, where what we are doing is, we are taking the alanine, going to the liver, we are coming back in the form of glucose. So this entire thing is referred as the glucose-alanine cycle. This entire thing is referred as glucose-alanine cycle, or it is also called as Cahill's cycle. Glucose-alanine or the Cahill cycle. What is the most important thing in this Kyle cycle or the glucose alanine cycle is what they ask is what is the name of the transporter? What is the name of the transporter? The name of the transporter is alanine. What is the name of the transporter of ammonia from skeletal muscle to liver? The answer is going to be alanine. The name of the transporter is alanine. This is the most important thing that they ask. Okay, now the next thing is the second one. How we transport the ammonia from the brain? From brain, we can say like this: transport of ammonia from brain and most of the tissue. Brain and most of the tissue to liver. How we are going to transport the ammonia from brain and most of the tissue to liver? So to understand it again, I am giving just a simple diagrammatic representation. For example, let's say this is the representation of the brain and here is the liver. The brain and the liver. So how we are going to do that? For example, if these are the ammonia molecules which are there in the brain and we want to transport. So what we do is we know that we have an amino acid with the name of glutamate. This glutamate may club with the ammonia molecule and will make a molecule that is with the name of glutamine. Glutamine. When the glutamine is made, we require energy. We require ATPs for the synthesis of glutamine. Whenever you synthesize any molecule, just pay attention, see what I am saying is, whenever you synthesize any molecule, the name of the enzyme is either going to be synthase or synthetase. So when it is going to be synthase or when it is going to be synthetase, this is a thumb rule that whenever you are synthesizing a molecule and you are using energy, the name of the enzyme is going to be synthetase. And if you are synthesizing a molecule without energy, the name of the enzyme is going to be synthase. So here we are synthesizing the molecule glutamine with the help of ATP. So the name of enzyme is glutamine synthetase. Glutamine synthetase. So we are synthesizing with the help of the glutamate and ammonia will make a bigger molecule, that is glutamine, with the help of ATP. So it's called as glutamine synthetase. Now this glutamine can easily travel via the blood. Will travel via the blood, will reach here. Once the glutamine will reach here, we can easily convert back them into the parent molecules, that is the ammonia and the glutamate. Ammonia and the glutamate, right? So to do that, the name of the enzyme that is going to be used is the name with the name of glutaminase. Although the name of the enzymes are not very important for this particular reaction, the name of the enzyme is glutaminase. So this is how we are going to transport the ammonia from the brain and most of the tissue. Now what they are going to ask is what is the name of the transporter? The name of the transporter is glutamine. So glutamine is the transporter from the brain. From the skeletal muscle is the alanine, from the brain and most of the tissue is glutamine, and this is how it is going to be achieved. The reaction part is although not important, what is important is the name of the transporter. So somehow we have transported all the molecules of ammonia from the various extrahepatic tissue to the liver, and whatever glutamate we have sent to the liver, we have taken out the ammonia from that also. So basically we have loaded the liver with the ammonia. Okay, so what I'm saying is that what we have done in the last 3 steps is, if I just briefly revise them, first we have converted all the amino acids into glutamate. Then in the second one, we have sent that glutamate into liver and we have taken out the ammonia, that is called as oxidative deamination. Then in the step 3, we have transported the ammonia via various means, maybe via alanine or via the glutamate. So now the liver is rich with ammonia. We will try to convert that into urea. The next is step number 4. What we are going to do in that is, whatever ammonia we have made, we'll try to convert that into urea, and that is referred as urea cycle. This urea cycle is also called as ornithine cycle, is also referred as Krebs-Henselt cycle, So whether it is Krebs-Henselt or the ornithine cycle or the urea cycle, what is the ultimate agenda is that whatever ammonia that we have accumulated, we'll try to convert that into urea. And that is going to take place in the liver. That is going to take place in the liver. So hepatocyte is the place where we are going to have the urea cycle. If they ask in the hepatocyte, it is going to occur where? In the mitochondria or in the cytoplasm? To answer that question, what I can say is that urea cycle, there are total 5 reactions. In the urea cycle, there are total 5 reactions are going to be. Out of 5 reactions, 2 reactions are going to occur in mitochondria, and the remaining 3 reactions are going to occur in the cytoplasm. First 2 reactions are going to occur in mitochondria, and the remaining 3 reactions are going to occur in cytoplasm. So urea cycle is the reaction which partly occurs in the mitochondria and which partly occurs in the cytoplasm, partly in the mitochondria and partly occurs in the cytoplasm. If we see the reaction part, how the reactions are going to occur. So the first 2 reactions are going to occur in mitochondria. The first reaction is like that. First of all, whatever ammonia you have accumulated, it will club with carbon dioxide and will synthesize a molecule that is called as carbamoyl phosphate. It is called as carbamoyl phosphate. In the synthesis of carbamoyl phosphate, we require 2 ATPs that converts into 2 ADP. 2 ATP convert into 2 ADP. So we are synthesizing a molecule using ATP. So the name of the enzyme will be synthase or synthetase. Yes, it is going to be synthetase. So the name of enzyme is carbamoyl phosphate synthetase. The name of the enzyme is carbamoyl phosphate synthetase 1. In short, it is also written as CPS1, carbamoyl phosphate synthetase 1 or the CPS1. This CPS1 is considered to be the rate limiting enzyme, is considered to be the rate limiting step of urea cycle. Now, what is the meaning of rate limiting step? Rate limiting step means the slowest step of the pathway is considered to be rate limiting one. So this is the rate limiting step. This CPS1 is activated by a molecule. CPS1 is activated by a molecule, or I can say there is an activator of the CPS1 which can activate the CPS1. The name of that molecule is N-acetyl glutamate. N-acetyl glutamate. In short, that is written as NAG. So N-acetyl glutamate is the activator of the CPS1. N-acetyl glutamate is the activator of CPS1. Then this carbamoyl phosphate, what it is going to do, the second reaction that is going to again occur in the mitochondria only, that it is going to react with a molecule with the name of ornithine. There is a molecule, ornithine. This ornithine is going to react with the carbamoyl phosphate. Now what we do is, in the second reaction, this carbamoyl group of the carbamoyl phosphate, this carbamoyl group is going to be transferred on the ornithine. So the name of the enzyme is, because we are transferring the carbamoyl group, so the name of the enzyme is transcarbamylase. And we are transferring on the ornithine, so the full name of enzyme is ornithine transcarbamylase. Ornithine transcarbamylase. And the product that we are going to get is citrulline. So the first two steps, I am just giving the numbers for our convenience. This was the step number 1 and this was the step number 2. These first 2 reactions occurs in the mitochondria. They occur in the mitochondria of the hepatocyte. So if I just make a demarcation, these are the first 2 reactions that are occurring inside the mitochondria. So this is the demarcation for the mitochondria. So inside to this is the mitochondria and here is the cytoplasm. So the first 2 reaction occurs in the mitochondria. The first 2 reaction of the urea cycle occurs in the mitochondria. Then the citrulline will come outside in the cytoplasm. Then the citrullin will come in the cytoplasm. Then this citrullin, once it comes in the cytoplasm, what it will do is it will react with a molecule that is with the name of aspartate. So citrullin and aspartate will join. These two small molecule will join with the help of ATP. ATP will come into action. Adenosine triphosphate will convert into adenosine monophosphate, means triphosphate will convert into monophosphate, means 2 phosphate will be consumed, and citrullin aspartate will club and will make a bigger molecule, and that is called as arginosuccinate. That is called as arginosuccinate. So, if I ask you that what will be the name of enzyme, the two small molecule citrullin aspartate joining with the help of ATP, what will the name of enzyme? Is it going to be synthase or synthetase? Yes, it is going to be synthetase because we are using ATP. So, the name of enzyme is arginosuccinate synthetase. Arginosuccinate synthetase, and this is going to be the step number 4. Sorry, step number 3. So this is going to be the step number 3. Once you have made the arginosuccinate, it's a big molecule, it's split into 2 molecules, means it will undergo lysis. This arginosuccinate is going to split into 2 small molecules. One is called as fumarate, and the another one is the arginine. It is going to convert into fumarate and arginine. When the arginosuccinate splits into 2, fumarate and arginine, it is basically a lysis reaction, right? So the name of enzyme is arginosuccinate lyase. The name of the enzyme is arginosuccinate lyase because it is a lysis reaction, or in short it is also written as arginosuccinase. This is going to be the step number 4. So we are going to get the arginine. Then the last step is this arginine is further split into 2 small molecules. It's further going to split into 2 small molecules. One is the ornithine, and the another one is the desired product, that is urea. So arginine is splitting into 2 molecules. Again, it is going under lysis, but this lysis requires water. In this lysis reaction, we require water. So it is better to say it is not lysis, it is hydrolysis. So the name of enzyme is arginine hydrolase. Or it is also referred as arginase. Arginine hydrolase or arginase. So basically it is a hydrolysis reaction. So this is how the urea will be made. And this urea, where it will go, as I told you that this urea is supposed to travel all the way to the kidney, and via the kidneys this urea will be excreted out. This ornithine, what we will do with the ornithine is, this ornithine can be reused. And this ornithine molecule can enter this ornithine molecule. Okay, so what we have discussed is till now is we are in the urea cycle, and we are going to have the 5 steps. The first 2 we have did in the mitochondria, the remaining 3 we did in the cytoplasm, and at the end of the step 5, what you can observe is that we have reached to the product that we were willing from the urea cycle, and that was urea. This urea is a water soluble substance, so it will reach to the kidney and will be thrown out of the body via the urine. Now, what happens with the ornithine is, ornithine can be reused, and this ornithine will enter in the mitochondria. For recycling, what we need to do is, this ornithine can go inside the mitochondria, and for that we have a transporter. The name of the transporter is ornithine transporter 1. Ornithine transporter 1. With the help of that, the ornithine will go inside and will join the urea cycle and will be reutilized here. So this is how the complete urea cycle occurs. You can see that the first two steps occurring in the mitochondria, remaining three steps occurring in the cytoplasm. Ultimately, we are going to get the urea that will be thrown out of the body. So this is how the normal urea cycle occurs, and these are the four steps of the biosynthesis of urea. Just revising it again, first we need to convert every amino acid into glutamate, transamination, then the oxidative deamination, then the transport of ammonia, and lastly is the urea cycle. So what are the points that they ask in the exam from the urea cycle, and what are the clinical correlations that we should know? When it comes to the points to be remembered, the first is what is the rate limiting step? The rate limiting step is CPS1. Then what they ask is, who is the activator? Who is the activator of CPS1? The activator is N-acetyl glutamate. NAG is the activator of CPS1. This is first thing they ask. Then the next point they ask is the energetics part. The energetics of urea cycle means how much energy is required in the urea cycle. When it comes to the energetics part, what we can say that the total ATP required, if you see, there are total 3 ATPs are required. If I just mention the steps so that it will be easy for you to interpret the thing, you can observe that in the step 1 we have used 2 ATPs. In the step 1 we have used 2 ATPs, whereas in the step number 3, if you see the step number 3, we have used one more ATP there. So total we have used 3 ATPs. This is one thing. But in case if they ask, instead of ATP, what they say is total phosphate used or total phosphate required, then the answer will not be 3, the answer will be 4 phosphate. The answer to this question is 4 because in the step 1, if you see, if I just scroll up to make you understand, what you can see here is in the step 1 you can see that we are using 2 ATPs converting into 2 ADPs, triphosphate converting into diphosphate. When one tri convert into one di, one phosphate is consumed. Here 2 ATPs converting into 2 ADP means 2 phosphates are consumed. As well as if you see the third step, step number 3, here you can see that the ATP is converting into AMP, triphosphate is converting into monophosphate, again 2 phosphate are consumed. So total in all, if they ask that how many phosphate are consumed, then the answer is going to be 4. The answer is going to be 4. So ATP required 3, if they ask phosphate, it is going to be 4. The next thing they ask is, what is the formula of urea? What is the formula of urea? If I ask the formula of urea, the formula of urea is NH2CO NH2. Now what they ask is, from where these nitrogens are coming. See, the first nitrogen that is there, it is from the ammonia that we started with. If you see the first reaction, we started with one molecule of ammonia. This CO group is coming from the carbon dioxide. We started with one molecule of ammonia, one molecule of carbon dioxide. Then from where the second nitrogen is coming, from where the second ammonia group is coming. This second nitrogen is given by the foreign molecule that is joining in the step number 3, and that is with the name of aspartate. So in urea, what they ask is from where the nitrogens are coming. One nitrogen is by the ammonium ion and the second is by the aspartate. Right? The first is from the ammonia and second is from the aspartate. This is what they ask. Then the next thing that they ask is, and it's very commonly asked, that see, there are 2 reactions that is occurring in the mitochondria, 3 and they are occurring in the cytoplasm. What they ask is, all of the enzyme of the urea cycle are cytoplasmic except, means they will give 3 options from the cytoplasm, one will be mitochondrial, or we can say which one of the following is the mitochondrial enzyme. So you should be knowing which one is cytoplasmic, which one is mitochondrial, right? So if you observe the cytoplasmic enzyme, just scrolling up the data and you see the cytoplasmic enzyme, if you see the step number 3, the cytoplasmic enzyme that is there is arginosuccinate synthetase, then arginosuccinate ligase, then the step number 5 is arginine hydrolase. All the cytoplasmic enzymes are starting with letter or alphabet A. All the cytoplasmic are starting with the alphabet A. So let's write down this point that all the cytoplasmic enzyme of urea cycle starts with the letter A. All the cytoplasmic enzyme of urea cycle starts with letter A. So this is an easy way to remember that the options starting with letter letter A are the cytoplasmic enzymes. Then is the clinical part. What is the clinical importance of knowing all these urea cycle and all? See, if there is deficiency of any of the enzyme in the urea cycle, what is going to happen is that the ammonia is unable to convert into urea. If the ammonia is unable to convert into urea, the ammonia is going to accumulate, and that condition is called as hyperammonia. And as we all know that ammonia is very toxic and it will produce many clinical disorders. So what I can say is this group of disorders comes under the category of urea cycle disorders. Urea cycle disorders. Now, what is urea cycle disorder is, to make it relatively easy, we can say, what I can do is, see, we started with the ammonia and carbon dioxide. The first enzyme that plays a role was the CPS1. Then we have discussed about something called as the ornithine transcarbamylase. I am writing OTC, the second enzyme. Then there was the third enzyme. Then there was the fourth enzyme, and ultimately the last enzyme, and we reached till urea and ornithine. So this is a simple way to represent the urea cycle, and I have written the important names of the enzyme. The first two reactions are going to occur inside the mitochondria. So this much part is going to occur in the mitochondria. This is the mitochondrial part, and these are the cytoplasmic reactions. And what is going to happen with the ornithine? See, ornithine is going to be reused. This ornithine is going to come back and it is going to be reused somewhere here. The ornithine is going to be used somewhere here. When the ornithine comes back, we require a transporter at the mitochondrial membrane, and the name of the transporter we have written was ornithine transporter 1. So this is the basic outline of the urea cycle. So if there is deficiency of any of the enzyme, the ammonia will be unable to convert into urea. What we are doing here is basically we are converting this ammonia molecule into a non-toxic urea. If there is deficiency of any of the enzyme, the ammonia will not convert into urea. Say for example, if I say that there is deficiency of CPS1. Let's say if there is deficiency of, if I say there is deficiency of CPS1. If there is deficiency of CPS1, the ammonia will not convert into urea, and this is referred as hyperammonia type 1. This is referred as hyperammonia type 1. If there is deficiency of ornithine transcarbamylase, again ultimately the ammonia is going to accumulate, and this will be referred as hyperammonia type 2. This is referred as hyperammonia type 2. So whether there is deficiency of any of the enzyme in the entire urea cycle, commonly it is seen with the first two, the CPS1 or the ornithine transcarbamylase. If deficiency of either of the enzyme, the ammonia is going to accumulate, and that will lead to hyperammonia. So how we are going to differentiate that? See, hyperammonia, whenever it is going to be hyperammonia, the manifestations of the hyperammonia is going to be the same. Whether it is type 1 or type 2, the clinical feature of hyperammonia is going to be the same. The patient will have vomiting, will have lethargy, will have decreased appetite, or maybe refusal to eat. Mostly these hyperammonia occurs in the neonates, right? Because they are the congenital disorders, these enzymes will be deficit since birth, so the symptoms will start appearing just after the birth, soon after the birth. So the child, the neonate, will have vomiting, lethargy, there will be refusal to eat, and ultimately he might end up in coma, right? When you do the ammonia level will be on the higher side, so it is hyperammonia. Now we need to rule out whether it's type 1 or type 2. How to rule out that? So one way is to do the enzyme assay. The another way of doing that is hyperammonia type 2 is associated with something called as orotic acid urea. What I'm saying is, how to differentiate between type 1 and type 2, because symptoms are going to be the same. In type 2, additionally what you get is something called as the orotic acid also comes in the urine, and that is called as orotic acid urea. So let's write down this clinically important point, that hyperammonia type 2 is associated with orotic acid urea. Hyperammonia Type 2 is associated with orotic aciduria. This is the additional finding that you are going to see in the question. So if the question says hyperammonia along with the orotic aciduria, you will say type 2. If there is no orotic aciduria, only hyperammonia, you will say type 1. So it is very easy and straightforward. There is one more disorder that we should know in the urea cycle. Let's see in the same flow chart. If I say that this ornithine transporter 1, there is mutation. If there is mutation in the ornithine transporter 1, the ornithine will not go inside. If the ornithine will not go inside, the step 2 will not occur, right? So again, it will lead to hyperammonia, but this time it is referred as triple H syndrome. Instead of hyperammonia, it is referred as triple H syndrome. So what are the importance of knowing triple H syndrome and how we are going to figure out that in a given MCQ? See, triple H syndrome is an acronym. Acronym in a sense that each H will have a meaning. The first H stands for hyperammonia, and that is very obvious. That is very obvious because the urea cycle is not going to run, so ammonia is going to accumulate, so it will lead to hyperammonia. That is one thing. The second H, see, if you appreciate in this diagram, what I am saying is that the ornithine is unable to go inside the mitochondria because there is mutation in the ornithine transporter 1. So because it is unable to go inside, so it is going to remain in the cytoplasm unutilized and it is going to increase in the body. So second H stands for hyperornithinemia. The ornithine is going to increase because it is not going to be utilized. So second H, the first H stands for hyperammonia. The second H stands for hyperornithinemia. The ornithine is going to increase. The third thing, the third H stands for homocitrullinemia. Homocitrullinemia. Just write it down. I will explain from where this homocitrulline is coming. So triple H stands for hyperammonia, hyperornithemia, homocitrullinemia, and it is due to mutation of ornithine transporter 1. Right? So the first two are very clear: hyperammonia, hyperornithinemia. The homocitrulline from where it is coming. Just pay attention and see here. See, what I am saying is, if the ornithine transporter 1 is not there, what you can observe is that if the ornithine transporter 1 is not there, what you can observe is that the first reaction can occur easily. There is no problem with the first reaction. The first reaction can occur easily. Even the second reaction, the enzyme is there. The enzyme is there. We are talking about that the ornithine transporter 1 is not there. So enzyme is there. So instead of transferring the ornithine group, the carbamyl group on the ornithine, the ornithine group, the carbamyl group will be transferred on lysine and it will make a molecule that is called as homocitrulline. That is similar to citrulline, that is called as homocitrulline. So this is called as homocitrullinemia. That is called as homocitrullinemia. So triple S syndrome is due to mutation in the ornithine transporter 1 gene. So these are the three clinical correlations that we should know in the urea cycle. Type 1 due to deficiency of CPS1, type 2 is due to deficiency of ornithine transcarbamylase, and ornithine transporter mutation will lead to triple S syndrome. So this is all about the general part of the amino acid. We have discussed the entire general metabolism of the amino acid, how they are ultimately converted into urea, right? Thank you guys.